We have the pleasure of welcoming Professor Margaret Titus, Chair of the Wednesday session of Myosin Motors in Vitro and in cells. Thank you so much for joining us. Give us a brief explanation or an overview of the session.
So the session is going to focus on how one of the major groups of motor proteins, the myosins, functions within cells to generate cell migration, and also in the case of some of the myosins that will be highlighted in the session, generating the tracks that the myosins work on.
And really, why is it important to highlight this subject matter?
So the myosins have been studied in great detail at the molecular level and also the tracks they work on. And we know a lot about their physical properties, their chemical properties, their kinetics in a test tube. What we really want to know now is how are those properties used or translated to the cell, and how does the cell generate movements using these various motors and associated tracks that they run on.
So what do you want attendees really to take out of this session? Because it is a very important topic to discuss.
So we're going to have 4 talks, and they're going to feature, 2 of the talks are going to feature the motor side of life, how the motors are working, and 2 of the talks are going to feature how the tracks are built. And so one of the talks is going to focus on how the motor works, what helps it generate its what's called the power stroke. And another talk is going to feature a group of actually ancient and conserved motors that they can motor, but actually they've been shown to help generate their own track that they walk on. 2 of the other talks are going to highlight how the tracks are generated, what are the regulatory factors that play a role in that generation, and then the nature of the tracks in the cell. So you don't have like a straight road running from point A to point B across the cell. You actually have this complicated network of tracks that the motors have to negotiate in order to generate movement within the cell. So those 4 things I think will be of interest to the broad membership of the society.
Fantastic. Thank you so much for explaining to us your symposium. Thank you for joining us, Professor Margaret Titus.
Thank you.